Good afternoon, everyone, and thank you for joining us for the second episode of Webinar Wednesdays with PKD Foundation Chief Scientific Officer David Barron. My name is Alexis Denny, and I work in the Research and Education Department here at the PKD Foundation. I'll be with you throughout the talk, so if you have any trouble, you can send me a message via your chat box or email me at education@pkdcure.org for help. I've also got Sarah Bristow from the Marketing Department here to live tweet throughout the event. Be sure to follow us at PKD Foundation and tweet her questions using the hashtag Webinar Wednesdays. She will add your questions to the queue to be answered at the end. We are excited to launch this new series as a way to educate you about the basic science of PKD. Each month on the third Wednesday, we will explore a new topic together. Today, we'll learn about cilia. The format for this series will be the same as last month and will include the presentation and then a Q&A at the end. During the presentation, please type your comments and questions into the chat panel on the right side of your screen or tweet them using the hashtag Webinar Wednesdays to @PKD Foundation. Please note that the PKD Foundation does not offer medical advice, and the information shared this evening is not intended, or this afternoon is not intended to be a substitute for consultations with your healthcare professional. Care and treatment decisions related to your health must be made in consultation with your physician or healthcare professional team. We are recording this webinar and will archive it to our website within the next few days. And now I will hand it over to David.
Well, good afternoon, everyone, and good morning to some if there are any on the West Coast. This noon topic is cilia. I don't know how many of you are familiar with this particular organelle, but I'll try to describe it. If you have questions, Alexis has covered that, but you can send a message via chat or the question and answer box or tweet them to @PKD Foundation using hashtag Webinar Wednesdays or email at education@pkdcure.org. So what are they? What do they do? And what can happen when they don't do what they're supposed to? I'm acknowledging a few of the sources where I've taken information from. These are in prominent journals, the American Journal of Physiology, New England Journal of Medicine, and Genome Medicine, one of the newer journals. And you can find information in a variety of sources if you're interested in further delving into the world of cilia. So what are they and what do they do? Cilia are motile, meaning they move. They're also called flagella, and they can propel individual cells through water, seawater, fresh water, and they can also be non-motile. In other words, they're just like little fingers, but very, very tiny fingers. And cilia and flagella are basically similar to cilia, but they tend to be a lot longer, have been around since single-cell organisms were the most advanced life form on the Earth. And curiously, they really haven't changed much since then, so they've been around for billions of years. Therefore, as is typical in biological deduction, they must serve some common function that's pretty important, otherwise they wouldn't have been preserved all this time. So if non-modal cilia are important, what is it that they do, particularly in the kidney? And in the kidney, it's the non-motile cilia that I'd like to talk about. But first, a description of cilia in general. As I mentioned, they facilitate the movement of single-cell organisms such as paramecia, which you may have studied in grammar school or high school. And importantly, they rid our lungs of mucus and all the pollutants that we breathe in, particularly if you're living in a big city. This is called the mucociliary elevator, a rather obscure term. But what it means is that these cilia are constantly moving what we inhale that doesn't belong in our lungs up and out so that we can swallow it and keep our lungs relatively clean. They also serve very important functions in reproduction, and they help propel the egg down the fallopian tube in mammals, such as humans, and they are the primary motor, so to speak, that allows sperm to swim towards the egg. So obviously, that means that motile cilia are very important just for the reasons of keeping our lungs nice and clean and facilitating reproduction. And here are a couple of pictures of these single-cell organisms, and you can see that the cilia are rather small, and the flagella are a little longer. And you can see how they work. They sort of rotate and beat, and it helps propel these single-cell organisms through whatever medium they happen to be swimming in. So motion is one important aspect of cilia. What do they look like inside? Well, I could spend probably the rest of the seminar and then a couple more included in describing this very complicated slide. But to the left of the slide is what this structure looks like in the electron microscope. And you see doublets, or two tubes sort of attached together, and there are 9 of these doublets, and they form a circle around a central pair of doublets. These tubes are called microtubules, which makes sense because they are very, very small. This whole structure is many fold smaller than a single hair in diameter. So we're talking about something that you could never see with the naked eye at all. And the structure internally, as an electron microscopist by training, to me is very beautiful. And you see in the color image how these proteins that make up the microtubules are designed. And if you see the little orange protrusions, that's a protein called dynein, which helps slide between these tubules such that the psyllium can wave back and forth. So that's what motile cilia look like, but many cilia don't move at all, except if they're moved by something. And it wasn't known until recently what exactly these structures did for most cells. It was originally thought that they might be vestigial organelles, sort of analogous to the appendix. Humans have an appendix, and it doesn't really do much anymore. It's a relic from when our diets included more raw foods and bones, and the appendix was an additional organ to help digest that sort of diet. However, that turned out to be the wrong thinking. And for something to have been conserved throughout evolution, they certainly, you would think, would be doing something. So what are the differences? They're actually not all that many, except that when you look at a non-modal psyllium in cross-section, and that's the right side of the slide, you see the same 9 pairs of microtubules. But if you look closely and compare it to the left side of the picture, they're not surrounded by dynein. And that's the little motor that helps the microtubules slide against one another. So without those little dynein arms and without the central pair of microtubules, the non-modal psyllium really just sort of sits there on the surface of cells. And where is it found? It's found in the kidney tubule, and that's what we'll be talking about today mostly. It's also found in the bile duct, and that's part of the reason that PKD also affects the liver. It's found in the pancreatic ducts, and in rare cases there can be pancreatic cysts as a result of PKD. And it's found notably in the eye, where it has to do with our ability to see, and it's also found in the ear and has critical functions in how we hear. And oddly enough, it's even in fibroblasts, which form connective tissue. And this was a surprise to me when I first saw a blunt little non-modal psyllium in a fibroblast when I was using an electron microscope. And I kept pictures of cilia because they kept appearing in the most unlikely places. And I just happened to have a hobby, or had a hobby, of collecting these photographs. So, so the primary cilium in the kidney is observed on the apical surface of the epithelial cells that line the tubules or the nephrons. So what is the apical surface? Well, apical in this case means the surface of the cells that are in contact with the fluid that is going to become urine. So these little appendages or little fingers are sticking out from the surface of these tubules, and the forming urine is flowing by them. So are they holdovers like the appendix, or do they really do something by sticking out there in the urine that's forming? And the big hint, obviously, is that they're not holdovers. Structures like this don't last throughout evolution if they weren't doing something significant. So what do they do? So this is a slide that takes you from left to right in a dramatic fashion, and I hope I can help you to appreciate this, that on the left is basically a whole nephron. You see a little colored, what looks like an arrow. That's the glomerulus, where the blood comes into the kidney, and the fluid comes out of the blood that is to be cleansed. And it goes into the proximal tubule and down the loop of Henle, back up into the distal tubule, and back down through the collecting duct. And after it gets out of the collecting duct, it collects in your bladder until you feel the urge to urinate. Now, taking that up many-fold in magnification, you can see what these tubules look like in cross-section. So that's in the middle of your screen on the upper side, and you see 6 cells, each with a single cilium, in the lumen of that tubule. In other words, these are hollow tubules and the fluid is flowing. Take that up in magnification a couple more times, and in the lower middle, you see a single renal tubule cell or epithelial cell, and again, that primary cilium is sitting there at the surface of the cell. You magnify that by thousands of times, and you see that these little finger-like appendages aren't so simple after all. They're only 1/10,000th the volume of a single epithelial cell in the kidney, but if you look what they do, they carry proteins up and down the cilium, and some of these proteins are critical to polycystic kidney disease. In particular, you can see little circles that are light blue and dark blue, and these are polycystin 1 and polycystin 2. And 85% of the mutations in autosomal dominant polycystic kidney disease occur in polycystin 1 and about 15% in polycystin 2. Those that happen in polycystin 2 are typically lead to a more mild course of the disease. And also, one from the bottom in that list, if you can read it on the slide, is fibrocystin, and that happens to be the protein that is mutated in autosomal recessive polycystic kidney disease. And the point that I really want to make here is that all 3 of these proteins that have mutations that are the cause of polycystic kidney disease are all associated with this non-modal cilium. So what is it really doing? Well, this is sort of a fuzzy slide, but it's to show a very simple point, that these little fingers that are sticking out in the forming urine are sensitive to the flow of that fluid. So if the flow is greater, or if the flow contains too much salt or too little salt, these cilia actually act as little sensory antennae that they can taste, so to speak, the fluid that's going by them. And depending on what they taste, they can allow calcium into the cell, and calcium is a signal for other things that happen in the cell. So this is a very simplistic view of what the cilia do, and I think this is the major take-home, that the cilia are critical to interpreting, if you will, what's in the forming urine and telling the cell to do something about it, either to conserve water or to conserve salt or to modify the urine in a myriad of other different ways. And this is a higher magnification and a more detailed magnification of what this cilia does. And I've highlighted PC1 and PC2 for the two autosomal dominant PKD proteins that are at the core of the disease. And you see a single cell, and where it says flow, that's the apical surface. And at the bottom of the slide, you see a wavy surface. That's called the basolateral surface. And that surface is separated from the apical surface by the little circles that you see on between neighboring cells. And the other point to this slide is not that everything needs to be explained, but the simple act of this cilium bending and responding to the flow of forming urine causes the influx of calcium into the cell, and that leads to a whole cascade of changes. And these changes, especially when there are mutations in these two proteins, cause abnormal cell proliferation. And that's something that leads to cyst formation. In other words, under normal circumstances, these cells would not divide, but in cells that have missing or mutated forms of these two proteins, the signal from calcium is to divide. And that's not what you want to have happen in polycystic kidney disease. So this is yet another cartoon of what's going on, and for those of you who learn visually, this is another perspective that shows you what's going on, that fluid flow on the upper middle left through the tubule in the kidney bends these cilia, and here they're shown rather exaggerated in size, but it proves the same point, that between polycystin 1 and polycystin 2 behaving normally, calcium comes in and the cell appropriately either absorbs water or absorbs salt or does what it's supposed to do based on the type of urine that's passing by. But when these proteins are mutated, the signal is aberrant. In other words, it's inappropriate for what the forming urine is. And this is what results in uncontrolled proliferation. Now, what's very interesting about the disease is that in most cases, even when these proteins are mutated, they don't proliferate abnormally because only about 1 in 500 to 1 in 1,000 nephrons actually form cysts. So it's still somewhat of a mystery as to what causes some cells with the mutation to begin proliferation and the formation of cysts and why others don't. It may be due to local injury that happens just like you scrape your skin. There are many hypotheses about that, and that's one of the active areas of research that's going on in PKD. So you can see by looking at two pictures, one normal and one with ADPKD, these are two cells next to each other, and you can see where they're attached. Normally they're attached pretty much near the top of the apical surface. Remember, that's the surface facing the urine. In ADPKD, the cells are misshaped a bit, and that junction at the apical surface is more towards the upper middle of the cell. And you can see that other forms of communication between the cells, ECAD, for example, stands for cadherin. And you can see polycystin 1 is also potentially on the basolateral membranes of these cells, but the interactions between the cells is different. Part B of the slide, which I'll show you in more detail, is what happens when you have a mutation and how things change. But that's a little too small to show you, so we'll go on to the next slide. And you can see that when the psyllium senses a signal but has mutated polycystin 1 and 2, that it sets off a whole cascade of events that lead to what you see on the right-hand slide. And this is very difficult to explain in an afternoon talk, but the circle that you see on the right of the slide is called the mitotic cycle. So M stands for mitosis, G0 is a cell that is resting and is not dividing. S, which is a big part of that circle, is when the cell is actively synthesizing DNA, because you have to double the DNA to divide into 2 cells. And all these things are happening much more frequently when polycystin 1 and polycystin 2 are not interacting correctly because of mutations. And if you see on the top of that circle what sort of looks like maybe a bird cage, that's really showing something called the metaphase plate. And this is part of the mitotic activity where the chromosomes are divided into each of the daughter cells. So when one cell becomes 2 cells, there's a very, what I consider, pretty phase of mitosis when you look at it from a structural perspective, where all the chromosomes are lined up in that line, and all the microtubules, which are not any different, curiously enough, from the microtubules that are in cilia, pull away these paired chromosomes so that each daughter cell gets a new set of chromosomes. Unfortunately, with this division, each new daughter cell also gets the PKD mutation. I also wanted to tell you why PKD1 mutations are so variable and more problematic. I purposely put big and little boxes around polycystin 1, or the PKD1 mutation, and a much smaller box around PKD2, which is the more mild form of the disease. And you can see that polycystin 1 is a very, very large protein, and it actually goes multiple times through the apical surface of the cell. You can see these loops, and you can see that there's also a huge amount of the protein that is sitting above the cell, that it's really outside of the cell, above the membrane of the cell. And then when you look at polycystin 2, it's really pretty tiny. It doesn't cross the membrane as many times, and there's really not much to it. It's a much simpler protein, but it's the one that allows calcium to go through, and it's the interaction between these two that really is responsible for the disease. So PKD1, in my experience, is one of the larger proteins around, and it makes the study of the genetics of the disease a real challenge, because there are mutations that occur virtually anywhere along the entire sequence of the protein. So from the membrane-bound part to all these various different domains, which have different colors and shapes, that are sitting outside of the cell. So what happens? Here are the 3 proteins, polycystin 1, polycystin 2, and the reason I showed you the former slide is because I wanted you to see truly how different they were. And here's fibrocystin, another ciliary protein that I mentioned earlier, that causes autosomal recessive disease. So when these cells, for whatever reason, through the cilium get a signal from calcium, they tend to form these cysts. And you can see this, that they occur many places in the nephron, and they grow and grow until you see a kidney that's virtually all cysts. And I think many of you have seen these types of kidneys that are often the size of footballs when a normal kidney is really the size of your fist. So this is really the core of the disease. Many strategies to treat the disease don't deal with these proteins specifically. They deal with downstream effects. In other words, you can try to stop the cell from dividing, even though it's getting a signal to divide, by diverting the calcium that comes into the cell. And there are various strategies to do that. Tolvaptan, which many of you have heard about, tries to keep the urine very dilute so that the cilium is less likely to let calcium into the cell. And that's one of the reasons why your physician may suggest that you keep yourself well hydrated. So what are the lessons here? Cells along the tubule look pretty normal, and that's what you're seeing on the very left-hand side of the slide. But some cells respond to a second hit, so to speak, and this is because the gene for autosomal dominant PKD dominates over the other gene from your parent, which doesn't have the PKD mutation, but it may get the mutation at random because mutations occur randomly every day in human beings, and that may be the second hit that causes the genotype. In other words, the cell has the gene and turn it into a phenotype, which is really what happens to you. And this is showing microscopically that these cells start dividing. They form an outpouching, which is in the middle of the slide, and that becomes a cyst, which eventually loses all contact with the tubule from which it came from, and these cysts become larger and larger. They result in fibrosis, which is the rat's nest sort of appearing around the larger cyst on the right-hand side of the slide. And sometimes they break down and release fluid, and that could be responsible for pain. They can also bleed, which is a serious complication. And inflammatory cells come in, and that's also leads to more fibrosis and potentially more pain. And we are currently supporting studies that are conducted in various academic centers around the country that are trying to get at what really is the second hit, and maybe even the third hit, that creates these cysts, which are so damaging to the kidney in some tubules, but not in all tubules. And what does this translate to? Well, actually, this slide is somewhat optimistic when you look at other kidney diseases. For example, ADPKD, which is the red curve, shows that GFR declines after the age of 30, whereas in type 2 diabetes, kidney failure occurs often much earlier. And rarer diseases like membranous nephropathy, IgA nephropathy, and forms of glomerulonephritis, where you leak a lot of protein into your urine, can lead to kidney failure even as a teenager. And there are other rare diseases like primary focal and segmental glomerulosclerosis, which can lead to kidney failure even before you become a teenager. So it's not to say that PKD is a milder disease than these other diseases. It's not. It's just that it has a longer course. So, as I mentioned, the cilia, to me, are the major target because that's where the mutations are. But there are a lot of other ways to try to prevent cyst formation. And that is to attack all the things that happen as a result of these proteins in the cilium that are mutated. So you can look to apoptosis, and many people you'll hear pronounce the second P, but that's not the case. And this is how cells program themselves to die. And this occurs during embryogenesis, and it also occurs in everyone all the time. Cells get old and they die by a very ordered process that doesn't result in an inflammatory process. And that's the way that you normally get rid of aging cells and replace them with new ones. There are also all these mechanisms that lead to increased proliferation. So there are many drug possibilities to decrease proliferation. But while you want to decrease proliferation in the cysts, you don't want to decrease proliferation in your gut or your skin. And that's why some chemotherapy for cancers have so many side effects. And you can also go after the cilium directly, as I mentioned, and you can also try to inhibit the fluid that is secreted into these cysts, which is part of the reason that they get so much bigger over time. So autosomal dominant polycystic kidney disease, as well as the recessive form, really lead a growing list of diseases that are related to ciliary defects. And that's why I could talk for many, many hours about what is now called ciliopathy, or the pathology of cilia. And this is really the result of the fact that they are so important. So that among the ciliopathies, leading the list, as I mentioned, are PKD, but there are some rare genetic diseases, von Hippel-Lindau disease, nephrothipsis, something I still can't pronounce, retinal renal syndromes, because as I mentioned, the eye have cilia, Joubert syndrome, Meckel syndrome, Bardet-Biedl syndrome, and orofacial digital and June syndromes. These are all defects of cilia. And fortunately in PKD, these effects on the eye and the brain are not features of PKD. So different mutations to different proteins in the cilium can lead to different types of diseases. These are not, I would stress, the same mutations that are responsible for PKD, but they are related to these non-modal cilia. So what are the consequences of cilia-related disease? Well, cystic kidneys is the one that I think we're all most interested in, but retinal degeneration is a ciliary disease. Anosmia, the inability to smell, is related to cilia, and I was surprised to hear that, but when you look at the olfactory nerve, they have ciliary sensors that actually are responsible for translating odors into signals into the brain. Liver cysts and fibrosis, unfortunately, are part of the phenotype of PKD, particularly in autosomal recessive PKD, where liver fibrosis early on in life can be the dominant presenting form of the disease, and the kidney disease may be less prominent and threatening early in life. Obesity and diabetes may have something to do with cilia. A very curious phenomenon called situs inversus, where the heart is on the wrong side of the body, again is related to cilia, because cilia are very important in embryogenesis. So in other words, as the fetus grows, cilia are responsible for how organs are placed in the body. There are diseases that lead to bone pathology. Mental retardation is a prominent feature of some of these ciliopathies because the brain ventricles, the part of the brain that holds your cerebrospinal fluid, also contain sensory cilia. Fortunately, this is not a feature of PKD. And finally, infertility, because as I mentioned at the beginning of the talk, both the movement of the egg and the movement of the sperm are related to cilia. So there are a wide variety of effects of cilia, and to put it on the flip side, without mutations in ciliary proteins, when cilia are operating as they should, you can see how many features of your physiology are controlled by these tiny little organelles that for many years no one really knew what they actually did. So in conclusion, little structures can have huge consequences. And it's important to note that a lot of the research that led to the conclusions about the importance of cilia in polycystic kidneys and other diseases was that when these discoveries about cilia were made, it wasn't necessarily understood that that research would ultimately give an insight into the pathology of polycystic kidney disease. So when you sometimes see research that sounds very strange, or why would you study a zebrafish, or that sort of thing, it's that kind of research that ultimately leads to some of the huge advances in understanding diseases like PKD. And I think we're entering that phase right now. So this little wisp of an organelle that's 1/10,000th the volume of a renal tubule epithelial cell that belongs to that little cilium has led to a renaissance in the understanding of PKD and how to attack the pathways that result from that cilium into new therapies to avoid the worst consequences of both PKD and many of the other diseases I mentioned. So that's my talk, and I just wanted to remind you I will be giving future talks. I want to talk to you soon about the ABCs of drug development, a very different topic than today, a little more applied, but to give you some insight, hopefully, into how drugs get developed, why they take time, and why they're often expensive. And I really want to point out an important evening webinar talk by Dr. Arlene Chapman, who recently moved to the University of Chicago from Emory University and leads their nephrology section in the Department of Medicine, and is a professor there. And to remind you of the PKD National Convention in 2016 in Orlando, June 24th to 26th. So stay tuned for updates. So if you have any questions, I'd be happy to answer them, and I'd like to thank you, as always, for all that you do for the PKD Foundation.
Well, if you have questions, please go ahead and submit them. This was a pretty intense topic, and David did a really nice job of explaining it. So if you don't have questions, that's okay. We're not getting many right now. I am going to ask David to talk a little bit about what he'll be doing next week. He's going to a conference that is all about cilia, and so if you don't mind, while everyone out there is thinking of all their great questions, and I see a few coming in now, tell us a little bit about what you'll be doing next week.
Yeah, in a way, I will probably give a quick update to this talk sometime in the future, because I will spend next week at a FASEB meeting. FASEB stands for Federation of American Societies for Experimental Biology, and there will be a whole week devoted to nothing but cilia. That's all day and part of the night for 4.5 days.
You know, David, you told us that you could talk at length on this topic, so I think you'll that opportunity.
I'll be able to listen for 4.5 days and try to distill that down into what's new and intriguing about cilia, because any workshop or symposium on cilia of recent vintage has included prominent talks on polycystic kidney disease. So I'm not going there to learn about paramecium or how eggs or sperm or the lung, although I'm interested as a biologist, but I'm really there to learn how polycystin 1 and polycystin 2 and the cilia work together to result in proper renal function and what happens when those proteins are mutated. So look forward to a few highlights from that meeting. I bet many of you can't imagine that there'll be that many talks concentrated on such a little organelle, but that's the way science is, and I think the psyllium is a real key to solving this disease.
And a shameless plug for the foundation, I want to mention that we did fund a portion of that meeting in order to ensure that PKD is a topic. So just shamelessly letting you know that we are on top of that and making sure that PKD is at the top of their list. Okay, so we have, this is an interesting question. Someone is wondering if there's any impact of diet on cilia behavior. So is there anything as far as diet goes that would impact cilia?
That's a good question, because intuitively, and I'm not speaking definitively now, but the urine is comprised primarily of water and salt. And by salt, I mean sodium chloride, table salt, but it also contains other salts, potassium salts, magnesium, phosphorus, calcium. These are all salts in solution. And there are also other molecules in the urine that are somewhat bigger, like urea, so-called middle molecules that are even larger. And it is thought that a lot of these molecules are related to your diet, so that a diet high in protein from meat may have add a different quality to the urine from a diet high in protein from vegetables. So protein source can have an effect on the urinary composition. Of course, the amount of water that you drink with your diet has a lot to do with the quality of your urine, whether it's light or whether it looks dark. If it looks dark, it typically means that you're not drinking enough. But you also get water from eating a lot of fruit during the day, or vegetables like green peppers. They all have a lot of water. There are some animals like desert rats and certain cats that drink virtually no water. They get it all from their diet. So that's a very good question, and some of these differences in your diet may indeed mean that these little cilia are picking up different signals depending on what you eat and drink.
Very good. We have a very nice comment to just let you know that your talk was fascinating. So thank you for that, for the person who put that, and I agree, and I'm sure many of you do as well. So let's see. Someone is asking about, I'm not sure if this is really connected to cilia, about the mechanism of pain when a cyst breaks down or when a cyst ruptures, I think is what they're asking. And I'm not sure that that's really on topic.
I'll give it a shot anyway.
Give it a shot? Sure.
You know, because the cilia are responsible in an indirect or maybe even a direct way in why some cilia burst. If those cilia are still causing cell proliferation and the cyst gets larger and larger, all the cysts that are forming stretch the capsule of the kidney. In other words, the kidney is encased in a capsule that's made of connective tissue, and it has nerves in it. So the mere pressure of those expanding cysts that can all be traced back to the cilia that are not translating the signal that they get properly because of the PKD mutation are growing and pressing on that capsule, and the nerves are recording that as pain. And when one of them actually bursts, they can leak that fluid, which may be irritating and cause pain, and they can also bleed, and that definitely causes pain because blood is an irritant, and the fluid in the cyst may be an irritant as well. And so people may feel on a daily basis sort of a dull aching pain, but if you feel an acute pain, it may actually be because a cyst gets so large that it bursts, or it bursts because of all the inflammatory cells that are in and around it, and some cells die and the fluid leaks out. So that I can relate that back to the cilia.
I have a feeling you can relate everything back to psyllium. Let's see. Oh, someone is just asking when the FOSSIP talk is. That's next week. What are the dates of that?
That's July 19th to the 24th, and it's in Snowmass, Colorado, where I'm seeing that the evening temperatures are in the low 40s.
Oh, so you're going to miss the 90-plus degree weather we've had here in Kansas City the last few days.
That's correct.
Lucky you. Okay, let's see. So, let's see, as the kidney becomes covered in cysts, and not just covered, but really throughout the kidney, as cysts start to take over and nephrons are destroyed, can the remaining nephrons in their cilia become overloaded?
That's a very good supposition. That's really what happens. Early in the disease, as the cysts are growing larger, they compress the normal nephrons, and despite being compressed, they don't die immediately. What they do is they send a signal that we're not getting enough blood supply, and that's because the cysts are compressing the arteries that supply the blood to the nephron, and that's why many people with PKD have high blood pressure. And they also do something called hyperfiltration. I mean, the kidney's basic function is to be a filter. It also does things with vitamin D, and it keeps your red blood cell count where it's supposed to be, but its main function is really to clean the toxins and excess salt from your body. So that continues because, you know, obviously living donors can do just fine on one kidney, and even with PKD you have two kidneys, and you start out with more renal function than you need to survive. So you can go for many years, even decades, with reasonably normal renal function before finally those nephrons that are working overtime, so to speak, hyperfiltrating, actually give up the ghost, so to speak, and that's when the disease accelerates.
Okay, great question. Okay, we have one more here, and someone is asking if there's a relationship between calcium and cyclic AMP.
Yes, there is, and that's one of the mechanisms by which the cell gets the signal to divide, and that gets back to the way that tolvaptan works by blocking vasopressin, which causes an elevation in cyclic AMP, and that is also a signal for cells to divide. So calcium, all these salts for that matter, sodium, potassium, magnesium, they're all critical for cell function, and that's why when you get a routine blood test, you'll see that those salts, or those ions of salts, are routinely measured. They're all very important.
And if you are interested in learning more about vasopressin, you can check out our website to view last month's Webinar Wednesday topic, where David explained vasopressin and talked at length about that. So that is all the questions and conveniently also all the time we have today. Thank you, David, for this talk and for the information you provided. And thank you to the audience for participating in the webinar and for your questions. The PKD Foundation appreciates the opportunity to provide you with the best possible education. I will be emailing out a quick survey to everyone who registered for this presentation. And if you don't mind, to just take a few moments to complete that. I appreciate your feedback, and we'll use it to develop additional resources, programs, and topics in the future. And when you complete that survey, if you have topics that you'd like to hear David speak about in these sort of basic science webinar Wednesday talks, or in the evening more clinically focused talks, please do include them so that we can keep track of everything that you are interested in learning about. If you have questions or would like to reach out to us directly, please email me at education@pkdcure.org. And a member of the education team will be in touch. On behalf of everyone at the PKD Foundation, thank you for joining us this afternoon, and we hope to see you next month. Have a great day.